It's really a pleasure to welcome Professor Amy Kuceyeski today as our speaker, who's a professor in statistics and data science at Weill Cornell Medicine in New York, where she leads the computational connectomics laboratory, and is interested particularly in understanding how the brain works in order to better diagnose and prognose neurological diseases and injuries, with a particular focus on machine learning and neuroimaging, which obviously fits very well with the interests of lots of us in these seminars. And so I think today you're going to talk to us about some new AI approaches to sort of fuse and map between different connectomes, which we are very excited about. And so without further ado, I will pass the floor over to you.
Great. Thank you so much, Sarah. And thank you so much for joining while you're on your leave. I appreciate you coming today. And Petra, also, thank you for helping to organize. It's a pleasure to be here. So as Sarah said, I'm going to be telling you about some of the tools we've been working on in our lab. But I always like to start with sort of getting perspective about the brain and how it's the most powerful learning system in the known universe, right? It's got these 86 billion neurons with 100 trillion connections between them. It's this massively complex system, and it has a really efficient learning, and you can imagine things, create complex and beautiful objects. But this sort of 2.5 pound lump of flesh is still quite a mystery, despite decades and centuries even of scientific study, which obviously is good because we would all be out of a job. But this very complex system, we want to study it not just to understand what makes us human, but to sort of figure out how the brain can be damaged and repair it after damage or injury when something goes wrong, and also to mimic its capabilities through artificial intelligence. I'm sure you all know this, but neuroscience basically built the blueprint for artificial intelligence. The first neural networks that were designed were literally inspired by neurons. That's why they're called artificial neural networks. But now AI is becoming one of neuroscience's most powerful tools. So it's going in the opposite direction, and it's helping us to sort of decode brain activity, map circuits, and even generate new hypotheses about how the mind works. And this emergence of AI in the past few to several years as a central tool in neuroscience is driven by the explosion of the availability of big brain data, right? So these recording capabilities that we have in recording in vivo brain activity in like an awake, behaving animal, for example, or even a human in MRI, fMRI world, as well as algorithm development and powerful compute hardware. These things have all converged to sort of feed into this explosion of AI in neuroscience. And at the same time, insights from neuroscience are feeding back into AI. So there are lots of people working on models of, for example, visual processing that mirror the hierarchy of the cortex, right? So kind of doing this mapping of the processing of the biological brain and the processing of an artificial neural network of images, to sort of look at how they correspond to one another. There's also this idea of this reinforcement learning algorithms that are inspired by dopamine and reward prediction. So the boundary between AI and neuroscience is really dissolving in the past few years. So looking at the direction of brain to AI, we know that modern AI is founded on sort of artificial neural networks, which were inspired loosely on how neurons communicate. And more recent work has used these principles of biological vision, including hierarchical processing of sort of the features of an image and parallel processing, so like the what and where processing, to improve computer vision algorithms. And there is some evidence that the more similar an artificial neural network is to a biological brain, the more robust it might be to something like adversarial attacks, which involve adding this highly structured noise to an image to sort of trick a computer vision system into classifying it as the wrong thing. And so we also have something like biological computers, which I'm not going to get into, but they're attempting to use actual neurons to perform computations, which is something that's interesting, but I'm still not convinced by. And sort of LLMs and large language models that we're all using these days are based on what we might consider uniquely human, which is written language. And going in the other direction, we have things like brain-computer interfaces, wherein neural activity is recorded and translated by AI systems to perform tasks like controlling a robotic arm, for example, or generating text output. This is especially important for people who have paralysis or something like ALS locked-in syndrome. We also use AI to predict how a brain will respond to a given image or video. So these are so-called encoding models. And then you can also use them to decode the brain or to reconstruct the scene stimuli from brain activity, right? So this is encoding and decoding in this intersection of neuro and AI. And researchers are even working on, you should look this up, pretty wild, reconstructing imagined stimuli from brain activity, which is pretty cool. And there's advances in that as well. And then, of course, we have AI-guided image analysis, where you have software tools that will process and extract these measurements of brain structure and function, for example, cortical thickness, or volumes or lesion masks of brain images. So that's something that I also use in my work. But today, I'm going to be telling you about this brain behavior mapping, which involves basically taking some measurement of brain structure or function and mapping it, often using AI tools, to some out-of-scanner behavior. And when I say behavior, some of my researchers, my fellow researchers get annoyed, but this behavior is an umbrella that can sort of encompass many different things, including things like cognitive scores, even diagnostic labels, outcome prediction, age, sex, all of these things can be loosely sort of included in this umbrella of behavior. So in brain behavior mapping, we first need to measure the brain, obviously, and there's lots of different work that's in different sort of scales of measuring brain activity, all the way from single neural recordings, perhaps using calcium imaging or a neural pixel, all the way up to what I work in, which is sort of the entire brain, looking at the entire brain as a whole using functional MRI. And then when I look at the sort of brain networks, for example, you can look at things like local connections or connections between different parts of the brain, different regions in the brain. You can also look at topological properties that are either regional or network level or at a global level. And then you can also sort of be at a different scale in time as well. So all the way from instantaneous fluctuations in brain activity, which is what we look at when we're talking about fMRI, versus development, so across a lifespan, how a brain changes. And then there's also sort of evolutionary scale temporal changes that occur in evolution. So the green circles there are highlighting what I'm going to be talking about today, which is whole brain fMRI that's measuring sort of spontaneous fluctuations over time within a scanner. And then once we have these measurements of, let's say, brain activity or brain connectivity, so I also use diffusion MRI to extract structural connectivity networks, which are white matter networks, so I'm sure you all know about it. So once we have these networks extracted, we can then use AI to train a model to predict some kind of out-of-scanner behavior. For example, if we want to identify if someone's going to develop dementia or have a learning disability so that we can intervene early. One important part of this is that you can also incorporate non-imaging information into these models that will help improve the accuracy of the prediction of the outcome. And this treatment information is important because it can be included to look at the impact of therapies. And this is especially important clinically because you might want to try to test in vitro, in silico, test different therapies to see which one might be optimal for a specific person. And this is a step in the direction of what we call personalized medicine. One criticism of AI, obviously, is that they lack explainability or interpretability, and in some cases, this may be true. But there are lots of advances happening in AI algorithmic development that allow us to peek inside of the algorithm to see what's important to these models. And that can allow us to point to potential causality, which can then be tested in prospective experiments, right? So this looking inside of the model is one of the primary things that I'm interested in, as well as having accurate predictions. So let me tell a little secret about myself. It's not a secret. I hate collecting data. It's just like I don't want to do it. I just want to process and like be on my computer. I don't want to talk to people. So the past decade has seen me just getting happier and happier as more data sets are made publicly available. They're also as a bonus, they're also large data sets, which is something that's important for when you're using AI and training AI algorithms. And so here's just a few of the ones that we use in our lab as well. Okay, so I talked about, I think you all probably have a basic understanding of diffusion MRI structure, functional MRI. how we extract networks, essentially chop up the brain into these parts using some kind of parcellation. And then you can basically reduce the brain network to a matrix, where the matrix tells you how, in the example of structural connectivity, how much white matter goes between the two regions, right? So that's what's included as an entry in the matrix. It's basically the amount of white matter connectivity between two different parts of the brain. And on the functional connectivity side, there's lots of different ways you can extract it, but essentially it's the coactivation sort of how correlated each pair of regions are, how correlated their time series are, right? So this is the functional connectome. Now, you might imagine that they'd be pretty correspondent, so things that wire together fire together, right? But that's not the case. If you take the upper triangular part of the structural connectome and the upper triangular part of the functional connectome and you vectorize them and you correlate them, you get a, correlation coefficient of between zero point 0.3 and zero point 0.4. So it's moderate at best, right? And this is due to lots of things. There's both limitations in both structural connectivity measurements and functional connectivity measurements. There's lots of noise in each of these measurements. There's false positives that occur in structural connectivity, for example. However, you also don't really want your functional connectome to be completely constrained by your structural connectome, right? Because then it's really hard for you to have dynamic responses to your environment. So if you need to, say respond to a ball that's flying at your face that your child threw at you, you want to be able to respond really quickly. And having this sort of dynamic functional activation that's not coupled and totally informed by structure is going to be helpful for you responding to your environment and having dynamic sort of cognitive processes as well. So there's a whole sort of area where people use different ways to relate function and structure. These include things like neural mass models, the virtual brain, so sort of from Victor Gersa's group. These are sort of sets of differential equations that tell you how brain activity changes over time, and you can couple them and link them using the structural connectome. There's other things like graph theoretical measures, looking at topology and sort of path distance and global versus local activation flows, things like that, which are really interesting. Then you can also just straight up statistically map them to one another. So just let's say using regression, you can try to predict one from the other using different properties that are extracted. And in a step in that direction, you can also use AI to do the same thing, right? So you can use machine learning or AI to map structural connectivity to functional connectivity. So that's an example paper from about 5 years ago now, where they basically took the upper triangular part of the structural connectome, fed it through some artificial neural network, and tried to predict the functional connectome. And then there's other things I won't talk about it today, but we also in my lab use something called network control theory. That's an engineering principle that essentially assumes that the brain is this connected system that has a has dynamics on top of it, and you can move between different activation patterns in the state space. If you're interested in that, you can always reach out to me, or I think there's some talks on YouTube, perhaps. All right, so today I'm going to be telling you about the Krakencoder. It's my favorite GIF of all time. Essentially, you'll see why in a minute, why it's called the Krakencoder. But the Kraken is also this mythical creature that has long arms and is an ocean creature. But in any case, one of the things that you all probably struggle with, as well as me, is trying to decide on what pipeline to use to process these data, right? So do I do global signal regression or don't I? Do I use partial correlation or Pearson correlation? Do I do deterministic? Do I do probabilistic tractography, right? So all of these different choices can go into extracting a structural and functional connectivity matrix. And you can also use different atlases. And does it matter? Turns out it does. So this is this really cool analysis where someone took a brain behavior mapping data set. This is in fluid intelligence. And they have each column here represents a different choice in the processing pipeline. Okay, so the first one is like which atlas they're using. The second one is which network, which metric they use, partial correlation, Tikhonov regularization, which space they projected in, and which classifier they use to map the functional connectome to the outcome. And each line represents a single model with a different set of choices, and the color represents the accuracy of the model. So you can see that there's lots of variability in the accuracy of the model depending on the choices you make, right? And so one of the reasons we made the Krakencoder was because we wanted to just use all of them. So we decided we were going to try to make as many choices as we could. We used, I think, three different atlases, three different atlases, two different kinds of chartography for structural connectivity, deterministic, probabilistic, and then for functional connectivity, we have GSR, not GSR, partial correlation, full correlation, and the different matrices, different atlases. So we have 9 different flavors of functional connectivity, 6 different flavors of structural connectivity for each person, right? And so what we want to do is allow us to use all of these flavors by making a joint autoencoder space. So it's basically each flavor has an encoding and decoding arm that allows you to go between the latent space and the full matrix space. And this latent space is only 128 dimensions. So it's really, really small. We toggled playing, we played with that a little bit, and it turns out 128 dimensions works pretty well, which is amazing considering some of the dimensionality of some of these matrices is like 40,000, the upper triangular part, right? And so what we did essentially was make these joint autoencoders an autoencoder for each flavor. And then we have this shared latent space where they all move through. Okay. And so it's called a Krakencoder because it has all these arms that are encoding and decoding with this like shared center. And also an autoencoder, Krakencoder. Okay. Anyway, it's a portmanteau. All right, so we train the Krakencoder by making sure that our autoencoder paths are accurate, right? So we want to make sure that if we take the same flavor, let's say functional connectivity, full functional connectivity with like a Shen 268 parcellation, we want to make sure when we project it into latent space and go back out to that flavor, we don't lose much information, right? So we want to make sure that's accurate. We also want to make sure when we move across different paths to different flavors that that's accurate, right? So we want to make sure that if we go from functional connectivity to structural connectivity, that structural connectivity that's being reconstructed through the latent space is also accurate. And I think the magic sauce that Keith came up with is that he wanted to take, so let's say my 9 flavors of structural and functional connectivity are being projected into the latent space, and Sarah's 9 flavors of structural and functional connectivity are being projected into latent space. I want to make sure that my 9 flavors are close together and far away from hers, right? So I want to ensure that my own connectivity measures are very close together in latent space and far away from everybody else. So it's this contrastive kind of learning in the latent space. And one of the reasons we did that was because we noticed that in a lot of the models that were mapping structural connectivity to functional connectivity, what they were doing was mapping to a very similar functional connectome for everybody, right? So it was like very similar to the mean. And if you look at how accurate, if you just predicted the mean and you look how accurate you are with just like a Pearson correlation of the upper triangular portion of the measured and true, you get a very high value, right? It's good for everybody, but it's not good for a specific individual, right? So we wanted to make sure that our latent space was preserving the individual variability in the connectomes. Okay, so let's get to what data we used. We used a Human Connectome Project, young adult data to do the training. We split out a training set and a testing set. This is about 1,000 individuals between the ages of 22 and 37. Interestingly, they also have monozygotic twins, dizygotic twins, and test-retest data, which is really nice because then we can look at some sort of benchmarks for how well we're doing. They have resting state diffusion MRI. And then, of course, there's the out of sample. We wanted to do an out of sample testing of the Krakencoder on completely held out data from different data sets. So we used the HCP DNA, which if you've worked with that before, you realize that it's a very different acquisition for the D and the A. And they were done on totally different sites than the original YA data, which was just done on one scanner type. Okay. So as I said before, we want to emphasize individualness. So how do we actually measure that and make sure we're doing well at individual predictions? And so what we use is this sort of identifiability metric. So here again, we have our setup where we have, for example, one input functional connectivity, where we take the person's functional connectome, we project it into the latent space, and then we get back out a predicted structural connectome. How do we check how well our predicted connectome is doing? Well, we basically take our measured connectomes, flatten the upper triangular portion, make a subject by edges matrix, and we do the same thing for our predicted. In this case, it's a structural connectome, subject by edges. Then we correlate them to get a subject by subject matrix, right? So this tells you how predicted the measured and true values are for that connectome. And now you want the diagonal to be the max, right? I want my predicted connectome that's coming out of the Krakencoder to be most correlated with my measured connectome for that flavor. And I want it to be low, kind of a lower correlation for the other people. So you can count how many times your diagonal is the maximum across the rows. And that gives you a top one accuracy. So what percent of the time am I matching to myself at the very first, very highest value? Now, I might not be the first, I might not be the max, but I might be the second highest value, which is still pretty good, right? So you can actually calculate a percentile ranking of the diagonal to get an average rank percentile. Okay. So at average rank percentile, the higher that value is, the closer to one that is, the better you're doing. Okay. So that's just what we have here. So we have the observed versus predicted. And then you have the observed versus predicted here. Average rank percentile is 0.84, where you have like sort of high values on the diagonal, but not exactly perfect, which is would be one if you have a maximum along the diagonal. Okay, so let's take a look at our results. Here we have on each row is a different flavor of functional connectivity. The first top 9 rows are functional connectivity, bottom 6 are structural connectivity, and then the columns are the output flavor, right? So you're coming in through one of those flavors of connectivity, mapping to another flavor of connectivity. So this is showing you all the pairs of connectomes and how identifiable they are and their predictions. And the first thing you notice is that there's a really high level of correspondence between functional connectivity flavors, right? So from mapping between function to function within the same person, you're getting really high identifiability scores. Also, the same case with structural connectivity, a little lower, probably due to its sparse nature, but you're still getting pretty high values. And of course, the harder one is mapping between the modalities. You get a little bit of a drop, but there's certain flavors where you have an average rank percentile of 99, right? So that's still pretty good. To compare it to something outside of that, what we have on the bottom part here of the matrix is how well I do in identifiability if, for example, I use my own data from a different point in time to identify myself, right? Because there's about 40 individuals in the HCPYA that have a test-retest scan. And we took those 40 individuals and did the subject by subject correlation matrix, and we counted how many were on the diagonal. And you can see the self-retest there does pretty well for a lot of the flavors, but it's still 99% ranking. So the function to structure mapping for for a certain pair of flavors is doing as well as if I take my own matrix from a different point in time and try to identify myself. Now, structure to function was a little bit lower, 98 percentile ranking, which is still pretty good. And that does about as well as if I use my monozygotic twin to predict myself. So it's doing pretty well. Compared to previous literature, as I said, we used two other models to compare, and their average rank percentile was 53, which is basically a little bit better than chance. So we're really doing a good job at preserving this individual identifiability. And I didn't get into it here, but we also analyzed sort of the mean squared error. So we wanted to make sure that the values themselves also were accurate. So we subtracted the mean and then look at the sort of mean squared error of the residual. And that was also pretty good. And we also looked at graph topology. So we wanted to make sure that the topological properties of the networks were also being preserved. And that's also the case. So they're doing as well as and better than other algorithms at preserving that topology of the system. Okay. So one of the benefits and one of the reasons for us to make this Krakencoder was that we wanted to discard these flavors and just get one representation, right? So here I'm actually showing you the average rank percentile of the identifiability when I take all of the flavors projections in the latent space and average them to get a single 128 vector. Then I go back out to all the other flavors. So I take that average and latent space and I go back out and predict the structural and functional connectomes. And you can see that I do a really good job, even using both fusion functional connectivity and structural connectivity, or just fusion FC or fusion SC themselves. So this sort of fusion representation allows us to take all of the modalities for function and structure, all the different choices in our pipeline processing, and compress it down into this 128 dimensions. Do the 128 dimensions contain interesting information? So we took that 128 dimension data, and then we projected it just using a PCA and a t-SNE, and you actually see that male and female are quite separated, right? We didn't tell them who were male and who were female, But somehow falling out of the data is that this latent space is holding information about the sex of the individual. It's also reflecting information about the age of the individual. So here you can see that the sort of darker colors, which are the younger people, are on the upper left hand, and then it has a gradient to the older people, even though this age range is pretty small. We also see that the latent space has monozygotic twins are almost on top of each other in latent space, even though, again, we didn't tell them who are monozygotic twins. But you can see that we have in the latent space, each dot is a person, right? This is just a projection of the data. And you can see the monozygotic twins are connected to each other with a red line, which sometimes is usually just a red dot because they're basically right on top of each other. That wasn't the case for siblings or dizygotic twins, but as we know, monozygotic twins should be more similar to one another. So can we use this information in the latent space to predict something else, maybe a little more interesting, like cognitive processes or cognitive scores? And we actually see when we compare the latent space, the Krakencoder's latent space to the observed functional connectome, so all of the observed functional connectomes we put into a model after doing some dimensionality reduction. And we actually see that the raw data does a little bit better at sex classification, a little bit better at age classification. But for the things that are more difficult to predict, like cognitive scores, we do get a boost in our latent space, having a slightly higher accuracy for that. Now, again, one of the things I'm really interested in is breaking open that model, right? So I want to find out like which networks are more important for predicting certain outcomes. And so the way we came up for doing this is sort of it's like a sensitivity analysis or an ablation study, but it's not really ablation. But here, what we did was we basically took networks of interest based on the Yale classifications. So we have these 7 Yale networks plus a cerebellum and subcortical network. And we basically keep those values in the rows that are corresponding to the regions for that network. We keep the rows and columns in and as they are. And everywhere else, we replace it with the population mean of the training set. Okay, so it's basically trying to fix all of the other networks' connections and just keep that one network's connections varying across the subjects. Then we put them back through the latent space and do the prediction, and you see how much drop you have in accuracy, right? And that tells you how important that network is for the overall prediction of the outcome. And so here you can see the top one is the sex classification, middle one is age, and then total cognition is on the bottom. And again, here the sort of higher values are corresponding to more importance of that network. So for sex, we see these sort of subcortical and control and default mode networks. For age, you see more importance for salience and ventral attention and subcortex. And then the total cognition looks like it's a bit more important for visual and dorsal attention. Now, there are some differences for structure and function, which is also very important to look at as well. So I'm not going to go into too much detail, but I want to emphasize that this is a way of sort of breaking open the brain behavior models and being able to isolate the networks that are driving the prediction in this Krakencoder model. Okay, so how good is our Krakencoder in out of distribution data? So we freeze the model that we've trained on people who are aged 22 to 36, and then we apply it to people out of sample. So this is in the 8 to 21 on the left, that's the developing data, and the aging data 36 to 100 on the right. And you see we do pretty well. So we still, even without retraining, we still do pretty well at preserving identifiability of our structural and functional connectomes and the across modality connectomes as well. And then, of course, the fusion does really well in identifiability, the fusion sort of flavor. And so one thing that was interesting was when we took the latent space and we looked at the latent space of our D, A, and YA, what we found was something that wasn't great, which was that the site was driving some of these differences, right? So this is a PCA projection of you can see this is the HCP-YA on the right is this dot, and the D and the A are over here in this long thing here. Interestingly, also, you can see male and female are still split, right? So the male are on the right side of the blobs and the females are on the left side of the blobs. So that's good. And also age is really preserved. So you can see PC2 is basically reflecting age. So that's great, but it's bad that they're separated, right? It's not good that the side effects are there, which is something that we all struggle with as well, right? So what we did was we retrained the Krakencoder by penalizing site prediction accuracy. right? So we want the Krakencoder's latent space to not be able to predict what site they came from. However, we still want it to preserve age information and sex information, which falls out later. So we basically have these levers that we can pull to reshape the latent space to not reflect site, but still reflect age. And so that's what we have here. We have the different weights on those different penalties in the loss function. And here you can see when you have connectivity, you have this unbalanced accuracy that's about here. And then as you increase the penalty on site prediction accuracy to reduce it, you see that it goes down. And the same for age. If you have, we have this sort of lever we can pull to emphasize age, and we can keep that as something that the latent space should be representing. Okay, so once we did that, I think we picked a W of 1 here, or no, 10, to make sure that the site classification accuracy was low, but the age prediction accuracy was still high. How well does our identifiability do? Do we lose anything? So here we have the change from the original Krakencoder to the new one that doesn't contain site information. And you can see the biggest difference here is about 3% drop. So it's not too bad, right? So we're still doing really well in our identifiability. Now let's go back to our latent space and see if our site information is there. It's gone. Okay, so now PC1 is showing you age and PC2 is showing you sex. It's really well defined, and it doesn't have that site information and the problem that we had originally. Okay, so this is sort of showing you how you can use this flexible latent space to not contain information that you don't want it to contain and to contain information you do want it to contain when you're doing brain behavior mapping. So let's take it out for a spin. Let's do sex differences across the lifespan in connectomes. So here we took all of the individuals from the D and the A, and we also had a third data set where we had the Human Connectome Project disease data set have some healthy controls in them. So luckily, it's the same acquisition as the D and the A in the age range of the YA. So it's really nice because you have this now continuous sort of same acquisition all the way from 8 years old to 99 years old, including that little gap for the Y in the YA age range. All right, so now we first see, so these are the functional connectivities only are on the left, structural connectivities are in the middle, and then both structure and function are on the right. Each of the x-axis ticks are the age bin that we did the sex classification for, and the y-axis is the accuracy of the sex classification. Okay, and then we have for each bin, we have 3 different models. We have an ensemble model that basically is a logistic regression of all, trained on all of the different flavors and then made into an ensemble, taking an average. So it's basically using the same sort of raw connectome information to do the prediction that the Krakencoder has access to, because it has access to all of the flavors. Then we have the frozen Krakencoder, the original Krakencoder in orange, and then we have a sex fine-tuned Krakencoder in this kind of mauve or pink color. So the first thing you notice is that the sex fine-tuned Krakencoder does better at sex prediction, which is great because that's what we want it to do. And an important caveat here is that the sex fine-tuning was done using a completely different data set than the one that the sex classification was done on. So we wanted to make sure there was no data leakage there. So you can see that the sex fine-tuned Krakencoder does better across the board. And then the second thing you can notice is that the sex classification accuracy is lowest for the prepubertal people, which is not surprising because their circulating sex hormones are lower. And then as they go through puberty, it starts to get higher. Midlife is pretty high for functional connectivity. It starts to drop off again after sort of what we would think of maybe as menopause for females, when sex hormones start to become sort of weaker in both sexes. But on the other hand, structural connectivity is lowest in 8 to 11, but it continues to peak in aging. So that it actually is the highest in these older age ranges, which is really interesting. And then the fusion function and structures is kind of reflective of both of those flavors. So let's break open the model, see which networks are driving sex differences across the lifespan. Again, this is a really complicated, really full figure, so I'll walk you through it. This is the functional connectivity model is on the left, the structural connectivity model is in the middle, and the fusion one is on the right. And then for each of these networks, these sort of functional networks, we looked at how important that network was across the different age ranges. So the colored bars are the importance across age, the different age models. And you can see that and the smaller the bar, the more important that network was, the less of a drop we had when we included it. right? So the first thing you see is the default mode network is really important for sex classification across the board, across all ages. The ventral attention and sort of control, these higher order networks in the functional connectome are more important for predicting sex in functional connectivity world. In the structural connectivity world, on the other hand, we have these lower order networks that appear to be more important, the subcortical and cerebellar. Also, the default mode network sticks out a bit too. And then if you sort of analyze over time the change in the network's importance, we actually see that the cerebellum isn't overall that important, but it does become more important over age. Okay. And the other thing is sort of the somatomotor network. It has this inverted U shape where it becomes less important in midlife, but it seems to be more important at older and younger ages. And similar with the control network, we have this drop off in importance in the structural connectome. So this is kind of a way of looking at how the sex divergence of the networks change over the lifespan, which is important if we want to understand sort of sex prevalence in different diseases and onsets of different neurological diseases and disorders. Okay, how am I doing on time? I'm great. Okay. So we have actually, this is very new work that my student Yunrei has been doing. And here we were actually looking at the HCP 7 Tesla movie watching data. And here we just took the frozen Krakencoder that we had in our original paper, and we took their functional connectivities from the movie watching data and projected them into the latent space just to see what they look like and where they were. So each point here is an individual, and the same individual has 3 points, one for each of these movies as they were watching them. And this is the PCA of the raw functional connectivity, and this is the PCA of the Krakencoder functional connectivity. So you can see that the movies are actually more kind of separate. They're a little bit more split. Even though the Krakencoder is not trained at all on naturalistic fMRI, it's still doing a pretty good job of sort of pushing apart the different movie paradigms. Oh, sorry. IT hates me right now. Um, sorry, uh-oh, did I lose my Zoom? Oh no, I'm here. Okay. IT
No, that
keeps
seems
telling
still working.
IT keeps telling me to update my computer and I just never do, and they are really mad at me. All right, let's get through this. So the naturalistic paradigm was pretty surprising to me because I didn't think that it would show anything, but you actually see that this is a matrix that shows the correlation. So each of these sort of squares is a set of people. And then across the squares are the same sets of people. So you can see that the diagonals here are a little bit higher, which means that the same person under different paradigms has a nice correlation in the latent space, right? So there's still identifiability across the movies. However, the movies themselves, which are along the diagonal and the rest conditions, are more correlated between the people, right? So my social network latent space representation is more similar to your social network representation than your inception representation, for example. So there is something about the movie that's driving the latent space representation, but there's also identifiability of the individual within that space. Um, and then, yeah, so I said the last bit here, I want to talk about natural lesion studies. So we do a lot of studies on individuals with stroke or MS that have these sort of acute areas of lesioning. And that can really shed light on how the overall brain network works, right? So if you have a lesion in a specific region and you have a very specific impairment, then that part of the brain must be important for that particular behavior, or that connection in the brain must be important for that behavior. However, it's really hard to do this advanced MRI in patient populations. And as we already know, I hate collecting data, and it's even worse collecting data in patients. And so how do we actually do these natural lesion studies if we can't do functional MRI or diffusion MRI in these particular patient populations? So a few years ago now, maybe over a decade ago, we actually came up with this idea of a network modification or NEMO tool, which basically allows you to estimate the structural brain network disruption using only lesion masks that are acquired during clinical MRI. So we have this basically, you just have to upload a lesion mask and an anatomical scan that's with it if you want to co-register to MNI space, or just an MNI space lesion mask. It co-registers into this database of tractograms that are from the HCP dataset. YA data set. And it basically looks at the streamlines that pass through the lesion and tell you where on the cortex or subcortex or cerebellum those connections are. Okay, so it's basically telling you an estimated disruption of the structural connectome. And we've used this in lots of different studies. Our most recent one was with the Enigma Stroke Consortium, and there were about 800 individuals with stroke that had lesion masks. And we had various metrics of lesion overlap and disconnection that we then put head to head in predicting motor outcomes. And we actually found that our NEMO tool, with its estimated sort of global level of disruption in the different parts of the brain, was most important for, had the best prediction accuracy for outcomes, motor outcomes after stroke. And then you can look at the parts of the brain that are important for doing that prediction as well. So you might see where I'm going with this, but you can also take that lesion mask, feed it through the network modification tool, get an estimated structural connectome, which then you can feed through the Krakencoder to get an estimated functional connectome. So follow me here. I know it's an insane idea, but you can basically take that lesion and get an estimated structural and functional connectome just from the lesion itself. And we actually showed in a paper in 2021 that those estimated structural and functional connectomes had better prediction accuracy than observed functional and structural connectomes from the actual patient themselves in predicting disability. So that's the one on the left there. This is in a cohort of MS patients. Now, I will say one of the caveats there is that the functional connectivity, the fMRI that we ran in the patients was like 5 minutes long. So it's really short. It's got like, big voxel sizes. It's what we could do clinically, which is great. But it's not the most optimal connectome advanced MRI, right? So that might be one of the reasons why it's not doing as well. And the same for the stroke subjects. I have it on the right there. This is 121 patients with acute stroke. And here we showed actually that the observed functional connectivity is in red. It does pretty poor at predicting motor outcomes versus the estimated structural connectivity in blue and the estimated functional connectivity in green. And similarly, for MS, we didn't actually have functional connectomes for these people with MS. We just had their lesion masks. And we do a pretty good job of predicting SDMT, which is a sort of measure of processing speed. And so there's some evidence that these estimated functional and structural connectomes can be predictive of some outcome. And I kind of think that perhaps one of the reasons is because the lesion is the cause of the impairment, right? And if you focus in on the effect of the lesion on these high resolution data sets where you can do these estimates of structural and functional connectivity, it might be better than taking an observed measurement in a patient population that's one, hard to do, and two, probably pretty noisy, right? So this could be an alternative, to collecting resting state and functional and diffusion MRI in patient populations. All right, I'll finish with just some generalities. So we know that the AI has limitations. It's only as good as the data it's trained on. If you put bad data in, you're going to get bad algorithms, garbage in, garbage out. There's also these, mentions of lack of mechanistic rigor, which I also agree with, but I think it has its place in doing predictions, and you can find smart and clever ways to sort of break open the model to look at the importance of different things, which you can then test in prospective experiments to verify. There's also this idea of obviously one of the biggest problems is overfitting, especially in neuroimaging data sets, which are kind of on the smaller end, lack of generalizability. So it's always good to try and test your data in completely held out data sets if you can to verify that they're working. And then obviously we've all heard this, AI can be inefficient. And it really, I think a lot of the future, especially in smaller data sets, is making compact models that can be more generalizable and less inefficient. And for the last 5-ish minutes, or I'll try to do it quickly because I know we want to get to some questions. I just want to highlight the applications of AI in women's brain health are very exciting for me. However, only 0.5% of neuroscience papers to date have dealt with things surrounding women's bodies, right? So things like menstruation, pregnancy, menopause, oral contraceptives, very small number of papers. And so the data just doesn't exist, right? So the data is not there. In these big neuroimaging data sets, people generally don't collect things like hormone levels or reproductive health history or menstrual cycle information. The HCP-YA did collect the menstrual cycle information. However, if you look at it, it's Gaussian distributed, which it should not be. Should be flat distribution. So I think people are just not reliable when you ask them when the date of their last menstrual cycle, right? So it's kind of sort of this not good data. We at the WBHI, along with Emily Jacobs and Nina Milan, are really focusing on getting this data collected, right? So if we can collect the data, then we can apply all these really cool AI algorithms to understand how hormones can impact the brain, because we know it's an endocrine organ. And so this was just in 2024. The first studies of their kind came out following females across their pregnancy, which were pretty cool, and show that this really complex event can affect the brain. It basically, you see this cortical thinning that sort of rebounds after birth. Sarah, you might want to look if you haven't looked them up already. They're pretty cool. They do rebound a bit after birth, but it seems like even years out, you have a sort of lower level, but it might not be bad. It might be actually a good thing, revealing some kind of plasticity event. In any case, I think it's really important for us to understand how these huge fluctuations in hormones that occur in pregnancy can impact the brain. And actually, my co-director Nina Milan has come up with this digital twin of a female's brain throughout pregnancy that can allow you to visualize how a specific person's brain can go through pregnancy. So, Sarah, if you have an MRI, you could put it in there. I think it's coming out soon. You could put your MRI in there and sort of toggle your switch along your weeks of gestation to see how your brain changes. And then, yeah, this is just like a little walkthrough of what you can do with the digital twin. So you can kind of toggle these different gestational weeks and look at how different parts of the brain change for you specifically. So menopause is also something that's particularly understudied. So this is a huge shift in hormones that happens to 50% of the population. Still don't really understand how it works and how the brain is impacted by it. Obviously, it is. We have a lot of reports of cognitive fog and issues with memory that occur during perimenopause and postmenopause. So we want to try to figure out what is actually occurring in this longitudinal sort of setting. And so they're enrolling this longitudinal menopause project is enrolling like 700 to 800 people. They're going to follow them yearly over time with these really densely sampled collections, cognitive scores, blood biomarkers, reproductive health questionnaires that they're developing in-house. To try and figure out how changes that occur in midlife can impact dementia risk at later in life. I'm also actively involved with Women in Data Science and Kaggle when they're doing these sort of Women in Data Science datathons, where we're looking at trying to get people involved that wouldn't normally get involved in sort of AI challenges. And we tried to sort of do something around women's health, but to be honest with you, there's not a ton of data out there, as I said. So the best we could do, which was still pretty good, was to look at sex differences in ADHD with functional connectomes. This was in last year. There were over 5,000 people that competed from 100 different countries. There was some prizes and things like that. But in a year from now, we're hoping to do one that's more centered around women's health with the WBHI Brain Bank that's currently being created. We also have a seminar series. If you are not signed up, you are welcome to. We have a monthly discussions of different things all related to women's brain health. There's all of our talks are on YouTube. And then I will stop there so we have at least 10 minutes for questions. But essentially, I wanted you to kind of get an understanding of how AI can really help us understand this mapping between brain and behavior. And I want to make sure that I acknowledge my funding sources and my lab as well. All right, good.
Thank you.